definition by non-invasive species, but basically what that refers to is invasive species. And invasive in the sense that they arrive to those new areas assisted by who? By humans, by human activities. It can be an unintended introduction or it can be on purpose. So there are the two options, okay? But the real way to call a species invasive species is because the species arrive there not by its own means. If a species arrives on its own, it's just natural dispersal, a natural dispersal event for whatever reason. But if it was assisted by humans, then it is considered an invasion. And then, of course, over-harvesting. Here we have a pie chart showing how mollusks are the ones that, in proportion, have suffered the most in terms of extinct species that have been recorded so far. Then you have fishes and birds, and then other groups of vertebrates. And here what you have is an old picture from the beginning of the 1900s of people working in a factory producing bottoms out of the shells of some of these mollusks. Okay? Are there any questions? No? Ten more seconds. Okay, anybody remembers what big clade of invertebrate animals are we reviewing? Where mollusks belong to? Well, that is within the mollusks, but the mollusks belong to a larger clade of bilaterians. Remember, there was 3 big groups of bilaterians. Correct? Lophotrochozoa. And anybody remembers what are some of the features of Lophotrochozoa that are unique to this group, that if you observe them, you know it belongs to this clade? It's 2 of them. A lophophore, which is what? Do you remember? Anybody remembers what a lophophore is? Yeah, it is like these extensions, like cilia, that are floating around in the water column that are going to collect particles from the flowing water, so the organism, the animal, can eat those food particles. And what is the other feature of the Lophotrochozoans that is unique to them and is present in mollusks? It's not, these 2 features are unique, to Lophotrochozoans, but if you don't observe them, it doesn't mean that it is not a Lophotrochozoans, okay? So if you observe them, oh, great, because now you know who they belong to.
Is it the larva stage?
Yeah. Do you remember the name by chance?
Is it TH?
Yeah, TH something. Trophocore larvae. And that one is a ciliated larvae, and it is present in mollusks and some other groups of Lophotrochozoans. But they are not always present, okay? And regarding this question, anybody remembers what would be the difference between mollusks, shells, and brachiopods? Because in mollusks, you include oysters, clams.
The shells open dorsally and ventrally instead of like up and down.
Yeah, how the hinge is a structure that allows the opening and closing of the shells. That is one feature. But one that is even more important, and it is related to the traits, the two traits that we just mentioned, that is present in one of the groups but not in the others.
Is it the encased inward exoskeleton?
Say it again?
The exoskeleton?
No. Again, the two features of the Lophotrochozoans that are unique to them is the lophophore, okay, and the trochophore larvae. So in this case, the difference between mollusks and brachiopods, in addition to how they hinge to open the two shells, is also the presence or absence of the lophophore. Okay, the lophophore is present in brachiopods, but is absent in other shells that belong to the mollusks. Okay? Questions? No? All right. So let's move then to the annelids. Annelids basically are segmented worms. Many of them are going to be marine, other fresh waters, and there are terrestrial species, and the most famous one that probably you know annelids for is the earthworms. And also they can vary in length. They can be really tiny, from a few millimeters all the way to 2-3 meters in length. And there are two main groups. Traditionally, there were 3 groups where they were classified, but that taxonomic classification was dropped, and now there are 2 main groups that classify the annelids, the Rantia and the Sedentaria. Okay? Now, the Rantias are mostly marine annelid worms, and many of them are going to be predatory, others are going to be grazers, and there are they have modifications in every one of the segments, as you can see here in the picture. They have these divisions or peripods that are like legs for walking, but in addition to that, the marine organisms are going to have these extensions, like cilia, that they help them in order to move around the environment, and also they have sensors that allow them to know if they are in an appropriate environment or not. And they live in the aquatic floor of oceans or fresh waters. Now, when we move to sedentarians, the name says a lot. They are not highly mobile, so that's one main difference between these annelids and the Rantians, like this one here. And sedentarians, as the name says, they are very, most of them are sessile organisms. They form these tubes, and from these tubes they make extensions in order to feed. And then 2 other that are not actually that sessile, but belong to the sedentarians, are the earthworms that you know from your gardens, and the leeches. Okay? Leeches can be really tiny. There are not that many leeches around here in the Americas. Where they are highly diverse and present is in Asia, in particular in Southeast Asia. And they have this special mouth where they have like this kind of knives to cut a little bit of the skin and start sucking blood. And many of them especially bloodsuckers are parasitic, but in addition to that, some of them can act as vectors of certain pathogens. Now, importantly, they have this substance that they inject when they are sucking blood from the host called iridine, and that iridine chemical is highly used in the pharmaceutical industry because it is used to numb your senses. That is the natural purpose for that chemical, so you don't realize that the thing is feeding on your blood unless you see it, okay? So that substance had been used and now of course chemically modified into synthetic products that act in the same way as this one. Are there any questions? No? Okay, 10 more seconds. Ready? Okay, earthworms are very important for terrestrial environments because they have this important ecological role. When they burrow through the soil, as they are consuming the material that they consume as food, at the same time when they are pooping, they produce these castings that are very nutrient rich and is very good fertilizer for plants. And that's why many people buy them in Home Depot or Lowe's to fertilize the plants. So that's why they are very important in terrestrial ecosystems. In addition to that, when they are burrowing through the soil, they help to move material, they reduce the texture and oxygenate the soils. So that's why they are very important. Now, in terms of their biology, most of them are hermaphrodites, but they don't do self-fertilization. What they do is cross-fertilization. They cross each other and then they exchange their gametes, and that's how they avoid self-fertilization, which, as you know, increases the level of inbreeding, which is not good news for any species in general. And there are a few species that do asexual reproduction by fragmentation, meaning that if you cut them in half, every half is going to become a full-grown individual in a few days. Here is something that probably you don't notice if you have ever grabbed one of the earthworms in your garden, but they have these tiny legs in each segment that you are going to be able to see in this video, and that's the reason why they can move in that way, and it gives a little bit of independence to each segment. So every segment has a pair of those. Okay, any questions? No? All right, so that was Lophotrochozoans. Let's move to the other huge one, the Ecdysozoans. And anybody remembers why Ecdysozoans are Ecdysozoans? We have mentioned that a couple times. You can read the slide. Yeah, so the key is that they have to molt in order to grow, okay? And that process, the technical term is ecdysis, okay? That is the type, some of you were concerned about the type of questions that are coming into the next exam, because there is a lot of information in this second section of the course with all the organisms that we've been reviewing. This is the type of question that you can expect, which is not very detailed, specific to a single species or a single group of animals, but to a very big trade of animals like the ecdysozoans, okay? And ecdysozoans are mostly two groups, okay? Nematodes and arthropods. That's another type of question that you can find in the exam, okay? Those are still general but important questions, yes?
So we're not gonna have questions that are like specific to like a nematode or any other?
Unless it is something that applies in a more general way to many other organisms. I'm not going to ask you, for example, what is the name of the feeding apparatus of a snail?
Okay.
Okay, that's useless, right? Okay. All right, so nematodes or roundworms, they are highly diverse and highly dominant in ecosystems, dominant in terms of the number of roundworms that you can find in 1 cubic centimeter or 1 cubic meter of soil or water, because there are aquatic and terrestrial species, and many are going to be free living, and they are very important for the well functioning of the ecosystem. And the other important thing is that many of them are going to be parasitic, and we will mention some of those parasitic species in a second. And also they can range in size from very tiny, 1 millimeter, few millimeters, all the way to, it says 1 meter in there, but actually the record of a nematode, a parasitic nematode, is 18 meters. Anybody can guess who the host of that monster could be? It's a huge mammal.
A bear?
A whale. Okay, so yeah, that number in there is not the largest nematode that you can find in nature. So they don't have a circulatory system, and they do a lot of their processing, metabolic processes, through the cuticle. They have a very specialized cuticle. I will not go into the details of that cuticle, but there are different layers of the epidermis on how these worms manage to do a lot of the metabolism through the skin. And one nematode in particular, probably you have heard of it, C. elegans, is a soil nematode that has become a model organism for molecular biology, genetics, and understanding many different things of human biology. I'm just going to, yeah, this is the development of the embryo of a nematode. And if you remember when we were talking about the development of the embryos in animals, they go through all these different stages until they reach the gastrula stage. And if you remember, there are two types of development of the embryos. Anybody remember the two main groups that differ in the development of the embryos? That can be another question in the exam. There are two large groups in how they develop the embryo, how they divide until they reach the animal. Yeah. Protostomes and deuterostomes, yes. Okay? So, and we humans belong to what? Deuterostomes. And what is the difference in the fate of the blastospore when there is a blastospore already forming the developing embryo? What is the fate of the blastospore in deuterostomes? What does the blastospore become into in deuterostomes and in protostomes? That's another main difference between those two different forms of development.
It becomes the anus,
Yes, for who?
the deuterostomes.
Yeah, deuterostomes, and it becomes the mouth in protostomes. Okay. All right, let's move. Okay, this is one famous parasite of mammals, Trichinella spiralis. And basically it's a parasite that the adults insist into the muscle tissue. And we usually acquire that parasite that is inside every one of these Norsin cells when we eat undercooked meat. And sometimes they even resist very well cooked meat. So you have to be sure that the animals you are consuming are produced in clean conditions. Otherwise, you can get one of these worms, and it's common to be acquired from pork. Now, these cells that you see here in muscle tissue, they are called Norse cells, and this is another kind of behavioral manipulation of a parasite into tricking the host into doing something that benefits the parasite. So the worm developing inside these Norse cells basically produces certain chemicals that tricks the immune system of the host into vascularizing the Norse cell. So a lot of veins are going to develop and are going to be feeding into the Norse cell. For what? To bring nutrients into the developing embryo, something that of course is not good for the host, but is good for the parasite. Okay? So that is like a general thing. There are many other nice, well, not nice, interesting features about the life cycle of this parasite, but you can learn that in a parasitology class. All right, questions? No? Arthropods. This is the largest group, the more rich in terms of species of all animals, including vertebrates and invertebrates. So the estimates is a lot. Probably we have described more than 1 million species, but the estimates goes several orders of magnitude above that. Now, you can imagine that you can find them all over the place. So there is always a species of arthropod in every environment that you can think of. And the main feature is that they have a segmented body, and the main diversification of arthropods came about in the Cambrian explosion. And one of the classical body shapes was that of the phylum. You can see here in this fossil all the different segments going from the head all the way to the rear back. This is like an ancient feature because now with more modern arthropods, you will see that there is a lot of modification of different segments of the bodies of arthropods, and they have been adapted for different purposes. We will see that in a second. Are there any questions? No? Ten more seconds. Okay. Questions? Okay. Now, as I mentioned, over time, many of the segments were modified for different adaptive purposes. We will talk about many of those. But the main reason for those changes in adaptations were different type of regulation of the same gene or family of genes. If you remember, I mentioned already that there is a group of genes that are well preserved across the tree of life in animals that are the Hox genes. Those are like master genes coordinating the development of an organism. And some of them can have the same Hox genes or the same number of genes, but they are going to end up producing different modifications in the different segments of the body. Everything depends on the species. And how that can happen? Through the epigenetic regulation of those genes and the proteins that the same gene is going to produce. Because remember, every gene does not produce a single protein. There are a few that they do that, but most genes are going to produce more than one protein. Everything depends on the developmental program that they are going to follow, okay? Now, one thing that was not clear within the animals, especially between the arthropods and its sister group, the onychophorans, which is this type of invertebrate shown in here, is that if the Hox genes were present only in arthropods, so that they modified the body plans only in arthropods, so it was something that was present in the ancestor of this sister group. And when researchers actually used fluorescent markers to see when the genes, the Hox genes, were expressed, they found that the onychophorans also express all those Hox genes, which are colored here in green and in orange, in different segments of the body, meaning that the Hox genes were already present in the ancestor of these two groups, okay? This is just an example of how you can use phylogenies in order to test hypotheses about when certain modifications or adaptations occurred in time. At least we know that these Hox genes were present in the common ancestor of these two sister species. Now, general characteristics. Now we are entering to the part where many arthropod species have a lot of modifications in the different segments. Here is the lobster, and you can see here that there are many segments, but the portion of the head, many segments have been modified, especially the anterior portion in arthropods. There is a lot of modification for sensory purposes and also for mechanical purposes. In here is the best example because you have the pincers to grab food, you have maxilla that have been modified to consume and crush food particles, and there are also the papilla and other sensory structures, depending on the species we are talking about, that can contain chemoreceptors, mechanoreceptors, receptors for different pressure to measure the pressure in the environment where they are living. So all this lives in the anterior portion, in the anterior segments that have been highly modified. Now, the thorax portion is the fusion of several segments, and usually in arthropods is where you are going to find the main legs that are used for locomotion, and usually are going to be either 3 or 4 pairs of legs. And then all the rear segments of the arthropod body plan are usually modified also for sensory, but mostly for reproductive purposes. Everything depends on the species that you are analyzing, okay? And here a short video for you to see some of the modifications of the anterior segments in the lobster. So one thing that I want you to notice is that you have different types of modifications. The clearest one when you have a lobster is the pincers, right? Those are for grabbing the prey that they are going to consume. But there are also different maxilla that have been modified in different ways. There are some that are like spoons, and there are some modified that are like, some maxilla that are like spoons with cilia in order to collect food particles and bring them into the mouth. And then the mouth is also another set of modifications. And so if you eat lobster at some point, get curious about the lobster that you are going to consume and just inspect the head. so you can see all these type of different modifications that have occurred in this species. Now, I will mention about some modifications also in blood-sucking arthropods like mosquitoes and biting midges, because even though they are this size, millimeters in size, there is a lot of modifications in the anterior portion of their body. Now, as we mentioned earlier, so they have the cuticle, also known as the exoskeleton, and is mostly made of the polysaccharide known as chitin, okay? We will see other organisms that instead of chitin, they are going to have the ones that we have in the nails. I just forgot the name. But there are this switch of the chemical, the polysaccharide, where the hard structures are made of. Now, we already mentioned this, but for you to keep it in your head, they do the diasis, which is shedding the exoskeleton for them to be able to grow. Now, that is very important when you put the perspective of evolutionary time, because arthropods were some of the first animals to colonize land. And they were able to do it successfully because they had these adaptations that were very good at preserving moisture, okay? And also to withstand the gravity pressure that is not present in water. So a lot of support was already present in the exoskeleton of arthropods. I already mentioned this about the highly modified portions and sensory organs in the mostly in the anterior portion of the animal. They have open circulatory system, meaning that they have a heart or a set of hearts connected throughout, like little pumps connected across the body, but they basically dump the hemolymph, which is their blood, into the cavity that is known as what? What is the name of the cavity in arthropods? Do they have a coelom or a hemocell? Or no cavity? They do have a cavity, okay. We are narrowing down to two options. What type of cavity do you have as a human? Or any of you? The diaphragm, yeah, is a muscle that is going to keep your organs. But what is the name of the cavity?
We have all the koala, we have all three
Yeah,
genera.
column. Okay, so it's a coelom. So if we have coelom and we are vertebrates and these are invertebrates, what do they have? Hemocell, okay? And what is the difference between a hemocell and a coelom? Why they are different? Well, you have the three types of tissue, either in hemocell or in cell. It doesn't matter, right? But the main difference is what? Why your internal organs are not just moving around in your belly, just floating around? What is holding them together? The mesoderm, right? So when you have a coelomic cavity, the mesoderm lines the internal organs. There is a tissue of mesoderm, muscle, lining the internal organs to keep them in place. They are going to move because there is a cavity, but they are going to have some support. In a hemocell, there is no such support. They have the three tissues, ectoderm, mesoderm, and endoderm, but the endoderm producing the internal organs don't have any support. They are just floating around, okay? But in a few invertebrate organisms that we have already reviewed, we have some, is mostly hemocell, but they also have a little bit of coelom. Anybody remembers what would be the example of an invertebrate having both types? They have shells. Mollusks. You remember the figures where we saw the internal portion of a snail and also the internal portion of an oyster or a clam? They have mostly the hemocell where everything is dumped, okay, all the hemolymph or their blood, and there are certain organs that are protected by the cellum or the mesodermic tissue that is going to give some support to those organs. Anybody remembers what organs are protected in mollusks or some mollusks? Okay. Who's responsible for pumping the blood? The heart. Okay, so the heart is protected both in bivalves and also in snails. But in addition, in snails, also the gonads are protected in coelomic cavities. Okay, so all right, let's move on. When we are talking about arthropods, most of the aquatic species are going to have gills for the exchange of gases, but the terrestrial species usually are not going to have the gills. Instead, they are mostly going to have this tracheal system and a bunch of openings across the body. I will mention that for gas exchange. Also, if you ever grab a grasshopper or a cricket, you can hold it close or use a magnifying glass, and you will see all across the body, along the body, little dots. Those dots are openings, basically, that allow gas exchange. That is the way that they do it mostly in terrestrial species. Although a few terrestrial species, because they live in moist environments, they can also have gills, okay? Now, main groups of arthropods is these three listed here. Chelicerates, which includes the spiders, the mites, and the scorpions. Then we have the myriapods, centipedes and millipedes, and the pancrustaceans, all the other arthropods that we will mention in a second. So chelicerates have modified the anterior portion of the segments into feeding appendages. But in addition to that, some of them have also modified some of the rear segments into stinging organs like the scorpions to inject venom that is going to paralyze the prey, and then they can feed on the prey. Now there is a group that is very ancient that was a dominant invertebrate predator early on that was called the Eurypterids. Okay, and that was back in the Cambrian, but most chelicerates went extinct, especially during the Silurian, the Devonian period, because mostly were marine. And remember those mass extinctions in the Paleozoic during the Devonian and the Ordovician was due to reductions in sea level. All right. So most of the chelicerates belong to the arachnids, and these are the three main groups. You have the scorpions, the mites, and the spiders. The spiders have a general body where you already know they have these 4 pairs of walking legs in the thorax. All arthropods are going to have the legs in the thorax portion, okay? Usually the abdomen is going to have other modifications, but they are not going to have legs. In addition to that, they have the pedipalps and the chelicerate. The chelicerate is the one where they used to inject the venom, and the pedipalps are the ones that they use for mostly for sensory and for breathing activity, for recognition of the opposite sex. Now, they have this special type of lungs called book lungs that they use to do all the gas exchange. And as you know, many of them produce webs. But the spider web have different uses. Some of them are for capturing food, others are for mating purposes, others are for the babies, to protect the babies. They also use the spider web to escape from predation. Probably you have noticed this, especially in the tiny jumping spiders. When they jump to escape a potential danger, they shoot a line to don't fall into any place where they don't know where they are going to fall. It's like a safe line that they throw. Now, of course, they also have parasites, and some of the parasites that infect them, especially trematodes. Anybody remembers what type of parasites are trematodes? Do they belong to what large group of parasites that have highly complex life cycles and they don't have cavities? They are also known as flatworms, but the large group is what? Anybody remember, just by chance? Platyhelminths, okay? So those that go into med school, remember those names because you are going to be dealing with those guys a lot, okay? So platyhelminths, and some species infect the spiders, and they also manipulate the behavior of the spider, because the spider, especially orb-web spiders, so you can see these beautiful webs that they build. Instead of building those beautiful webs, suddenly they become cocoon builders. They build these webs in order to build an incubating chamber for the parasites, so they can survive well. And then after the parasite closes from those chambers, then the spider becomes the food for the babies of the parasites. Okay, so there are a lot of weird stuff in the parasite world. Myriapods, you know this well. There are, especially the millipedes are active right now. They are mating. So if you are taking a walk, probably you have run into some of those on the sidewalk. And they are, of course, they don't have 1,000 pairs of legs, but they have many. And they are mostly detritivores. So they consume a lot of decaying food particles that they find on the forest floor or on the gardens. And they are not dangerous at all. The ones that you have to be careful about are the centipedes, because they are predators, they are carnivores, and they have modified chelicerae at the front where they inject venom. Usually they are not that big, but it is painful if you get bitten. And in the Galapagos, there is a centipede that is giant. It's like this size. So it's, yeah, those ones are scary. But, yeah, and then the other difference between these two groups is the number of legs that they have per segment. The centipedes are going to have 1 leg per segment, whereas the millipedes are going to have 2 legs per segment. Okay? Now let's move to the rest of the arthropods, pancrustation. And in terms of phylogeny, the most recent evidence shows that insects are more closely related to a group of crustaceans, and actually is not the main group of crustaceans that is the most common one, like the lobsters. It's another group of crustaceans. There is still a lot more evidence that is needed in order to be completely sure about this relationship. But in any case, so this group of insects and crustaceans is the group that is called pancrustaceans, okay? So insects are more closely related to crustaceans than to myriapods and the chelicerates. And who would be the basal clade or the basal group in this phylogeny? What is the basal lineage in this phylogeny? If that is a question in the exam, you are given this phylogeny, and I ask who the basal lineage of this arthropod group is?
Would it not be the shell series?
Yeah, chelicerates. Okay, that's the earliest group of this monophyletic clay. Why do I say that they are a monophyletic clay, the arthropods?
Because the common ancestor and then all of their lineages. are
Yeah.
present.
So you have the common ancestor and all of the descendants. Okay. Okay. I have mentioned many of the information that is there. The only thing that I haven't mentioned from there is how they get rid of the waste material. A lot of the arthropods, the nitrogenous waste, is through the cuticle. But depending on the species, they can have some glands that they use to osmoregulate and get rid of the waste material. I already mentioned this. Basically, it's just a summary that the anterior portion is highly modified for sensory structures and for mechanical purposes. Then you have 3 or more pairs of legs in the thoracic segments that have been fused. And then the swimming appendages are in the abdominal segments, and also the modifications in the very rear segments of the body plan, mostly for breathing purposes and for sensory purposes. And here is where we start to see a lot or most of the species with separate sexes. Okay, so they are going to start having this nice feature for biologists in order to understand evolutionary purposes, because it is easier to understand when a species is a real species versus another one that looks very similar. Anybody remembers what is the species concept that we would use when we have a species that have sexual reproduction with separate sexes? The biological species concept, okay? And when Linnaeus started to describe a species, what species concept was he using? The morphological species concept. Now, if I tell you, okay, we have now a species that can only do a sexual reproduction, what would be from the concepts? We reviewed 3 concepts in the evolution section. What would be the concept that you would use? We already mentioned morphological, biological species concept. What would be an appropriate species concept for a species that doesn't have sexual reproduction? Ecology can play roles in many different concepts, species concepts. So what we just reviewed in the previous slide, when I was asking you about the relationships of insects, crustaceans, chelicerate, what was the drawing? How do you call that drawing there? Yeah, a phylogeny, right? So, phylogenetic species concept, okay? So, why is it useful when you don't have sexual reproduction? Because then you can use the evidence, all available evidence you have at hand, in order to see what species are more similar to each other. And especially it's useful when we use molecules or genomes, etc., all molecular markers. Because then some things that morphologically seem the same, when you put the molecular evidence, you see that they are not that similar. They are not the same species. They belong to different species. They might be sister species, but they are not the same. In that case, you don't have sexual reproduction, but you have enough information to tell them apart. Okay? within arthropods, the largest group of crustaceans are isopods, and many isopods are going to be parasites. I will mention a few in a second. Then we have the decapods, which include many of the things that you have for meals, like shrimps, crabs, lobsters, etc., etc. And the cuticle in many of these, we already mentioned that arthropods have the polysaccharide chitin, but in addition to that, they can have an extra hardening by calcium carbonate, okay? Which is also something that we have in our bones and in cartilage. We will talk about that in the next chapter. Most are going to be marine or fresh water, and a few are going to live on land. Many are going to be planktonic, so tiny, and many of those tiny arthropods are very important to sustain marine food webs, because they are the main food for large animals like whales or like the shark whales, that they consume krill. And if you've seen krill, it's like that one there, and that fin looks very large. They are really, really tiny shrimp-looking arthropods. Then we have barnacles, which are mostly sessile. You know the barnacles because they attach very hard to the hulls of boats, and basically they are protected by these shells, and the animal is just coming out in order to grab food from the circulating water. And one of the things that I wanted to mention before moving into the insects is that there are many parasitic species in barnacles and also in the isopods. One example in the isopods is this isopod that looks like a little crab, but they have a lot of modifications in the legs, and the legs are present in every segment of the body, and they have these very strong claws. Okay, when they are larvae, they are free swimming, and they are going to infect the next host, usually a fish, in the gills. So they are going to reach the gills, then they are going to mature, and they are going to mature in a different way. They have an interesting maturation cycle. First, they are males. Once they keep growing, they become females. And then once the female is inseminated with the male sperm that was previously there in the organism, they are going to move into the mouth through the gills chambers, the openings. They move into the mouth of the fish, and with these claws that are all across the body, they are going to grab the tongue of the fish, and basically the tongue is going to die because they cut the circulation and they become the tongue. So there are some freaky pictures of a fish opening the mouth and the little thing is just there. I mean, the fish is alive and it can still eat, but of course there are detriments to the health of the fish. So that is one example of a parasitic isopod. Another even weirder one is a barnacle, and there is a species of barnacle that infects these shrimps, and they are like an alien thing. They release these stages that are called the kertrigon, and the kertrigons are males and females. The female is going to mature and is going to release a structure that is going to inject something into the next host. So that structure is like a dart, and the dart is known as this spermigon, if I remember well the name, and it's going to be injected into the crab. The male is going to mature independently of the female in a free living way, and then this spermigon enters the crab, and then is going to go into the gonad tissue of the crab, is going to produce a castration of the crab, and also is going to mess up the hormonal system of the crab, and is going to, males are going to be feminized and they are going to become females. And why is that good? Because it allows better production of more parasites, more baby parasites for the next generation. Once that is mature, the female is mature, it develops a chamber where the male is going to lodge inside that chamber, and then they undergo sexual reproduction. and then they continue the life cycle. But it's a very complex life cycle. There are more details into that life cycle, but I'm just giving you the more general thing about that, that parasite. All right, questions? No? Let's move into insects, or also known as hexapoda. And they are all over the place. Any type of environment you will find them. And basically we have three segments: the head, the thorax, and the abdomen. That's the classical example. They have three pairs of legs. And in what segment are the legs in arthropods? In the thorax, okay? Always in the thorax. There are no walking appendages in the abdomen of insects, okay? There can be in other arthropods, but not in insects. And then they are going to start having a more developed nervous system. They have the ganglia in the anterior end, and then, well, in the case of grasshoppers, they have this nerve ring, and then they can have this extension in the lower portion of the abdomen that goes across the whole body, and there are some other extensions that go across the body to move the muscles that they have. Now, all this fossil is over 400 million years old for the insects, and the rapid diversification of insects occurred first in the Cambrian explosion, but then there was a higher diversification later on in the Paleozoic from 350 to 250 million years ago. That is about between the Carboniferous and the Permian periods, before the end of the Paleozoic, and they were the first ones to take flight, to exploit the niche of the aerial environment, okay? And of course, exploiting that niche opened the opportunity for further speciation of this group of organisms. Now, one important thing is that the wings of these animals are extensions of the cuticle, so they didn't have to elaborate anything anew, sacrificing any other segment from their body. They are just extensions of basically the skin that they have. This is one important feature that has been bursts of diversification of insects is what happened by the end of the Paleozoic, especially during the Carboniferous period, where it was highly humid, a lot of wetlands, because the gymnosperms appear. Remember, gymnosperms are older than angiosperms, okay? The gymnosperms appear as new evolved plants, and they open a new niche. They were plants with seeds, okay? That was a new niche that was exploited by many insects. And the second strong burst of diversification of or radiation of insects came about by the late Mesozoic era of the dinosaurs, by the early Cretaceous, late Jurassic, and throughout the Cretaceous, when we have flowering plants appearing in nature, okay? So those two periods provided a lot of diversification, a lot of new species of insects that are associated with plants. Now, in terms of how they develop, there are two ways that insects develop. Incomplete metamorphosis, like the case of grasshoppers. Basically, what you can see after the eclosion of the eggs of a grasshopper are like mini-me's of the adult, okay? So basically, that's all they do, and that's what we call incomplete metamorphosis, and the more technical term is ametabolous, okay? Now, complete metamorphosis, the classical example, you know, is the butterfly. They undergo all the different stages, so they have the larvae, which is the caterpillar, that is just a feeding machine. Then it produces the pupa once it has acquired enough food, and then the pupa inside develops the adult, which is the butterfly or a moth. So complete metamorphosis, or holometabolous. That's the technical term for complete metamorphosis. I'm going to jump this video, you know those. I already mentioned about the separation of the sexes, and many are going to have internal fertilization, although some insect species is just going to be external fertilizations. And some species are going to have elaborate copulation behaviors, and others are just going to just exchange the gametes, and that's it. And usually they are oviparous, so they produce eggs. Now, here is in insects the three main clades. So we have the most basal one, Archaeognatha. Then we have the sister one to the winged insects, the Signetomata, and probably you've seen these ones, we will see in a second. And then we have these four main groups with complete metamorphosis that include the beetles, Diptera or flies, Hymenoptera, and then the Lepidoptera. And then incomplete metamorphosis includes the Hemiptera and the Orthoptera. So very quickly, the most basal lineage, the Archaeognatha, has about 350 species. They are wingless and they need moisture in order to conduct their life cycle, and they are mostly detritivorous, okay? Also, but they are also herbivorous. Now the other one, Sigmentoma, about 450 species too. They are small, wingless, and probably you've seen them in apartment buildings. So they are these arthropods that they are very quick, so they move very fast. Probably you've seen them crawling around the houses or in apartment buildings. So they feed on decaying material. So they can infest buildings, which is not good news for builders because they need to do some spraying because these things eat the wood. Okay, so on any decaying material that is in the building, so they can deteriorate the constructions. All right, Coleoptera. Remember, all these next four groups have complete metamorphosis. Coleoptera is the most species-rich group of all the insects. Over 350,000 species have been described. 2 pairs of wings. The internal one is the one that actually they use for flying. It's a little bit flexible. And then the hardened portion that protects the other ones, the membranous ones that are in the interior portion closer to the body. And they have many modifications of the exoskeleton. Everything depends on the breeding behavior. As you've seen, many beetles have these huge horns that they use for breeding rituals and some mating purposes. And other species don't have that many modifications. Diptera is the second largest group of insects. Over 150,000 species have been described. The main feature of these ones is that they have one pair of functional wings, and the other pair has been reduced into 2 tiny kind of racket-shaped wings that are known as the halters. And the halters is just to give balance when they are flying. And many modifications, some of them are parasitic, or they are also known as micropredators, like mosquitoes, biting midges, black flies, etc., etc. And there are many free-living species. Hymenoptera, another huge group, over 125,000 species described. These include the wasps, the bees, and they are also known as the eusocial insects. Have you heard of that term, eusociality?
They're not social, or they just hate everything?
No, they are social, actually, but it is known as eusociality because they have this very specific system in how they are arranged. So they have different castes. Okay, so they have the queen, they have the soldiers, they have the workers, and there are other variations depending on the species. They build these colonies, and also there are some things about the genetics of these organisms. Anybody have heard of what is special about the genetics of the Hymenoptera? No? Usually the workers are haploid, and the only diploid is going to be the queen. And it's going to be only fertilized by a special cast of soldiers within the colony. Okay, but the female is the only one that is diploid, and she can produce diploid females or haploid males. Okay, so that is a special thing about the genetics of the Hymenoptera. Then we have the Lepidoptera, over 120,000 species. Mostly are going to be nectaritors. Okay, so they are going to be pollinators and nectaritors, moths and butterflies. And one feature about this group is that the wings are going to be covered with the scales, and the scales are protective, and also they vary in coloration, and coloration, depending on the species, is going to have relevance for the breeding system of the species. Certain type of mating behavior that the males or females are going to have, depending on the species. Now let's move into the two groups that have incomplete metamorphosis. The first one is the Hemiptera, over 85,000 species. Here is where we find what we call the true bugs, and many of them are going to be vectors of parasites, or they are going to be micropredators and ectoparasites. The most common one, when they infest dwellings of people, is the bed bugs. Okay, and the other ones are the assassin bugs, like the one shown in there. Assassin bugs can be parasitic on plants because they suck up the hemolymph, not the hemolymph, the sap of plants, or they can be parasitic in animals. There are some that suck blood from humans and from many other vertebrates, and these some species of this group are the vectors of what disease that we have mentioned earlier? A kinetoplast that creates a lot of troubles in humans, and one of the symptoms is an enlarged cardiac muscle. It's a trypanosome, and what is the name of the disease that it produces? And in what continent? Anybody remembers? Yes, it's a trypanosome, you're right. Chagas. The name of the disease that it produces is Chagas. Okay, and that one is restricted to what continent? The Americas. Okay. Well, not South America. We have it here in Florida, and actually, we with a friend here in public health, we are writing a grant trying to convince people to give us money to study this thing in Florida, because we have the trypanosome, which is Trypanosoma cruzi. We found it in insects. We found it in reservoirs, meaning cats, dogs, armadillos, possums, whatever thing. I don't remember. There is another main reservoir. Raccoons, we also have it in raccoons, and there are other species like some species of mouse. And there are human cases, but all those human cases recorded here in Florida are imported, are from people coming from other parts in the tropical Americas. But we have everything that we need in order for local transmission to occur, but we are not sure if local transmission is occurring and why it is occurring. So it is here in Florida, and especially the problem is in more rural areas, peri-domestic areas, where people come into contact more with synanthropic reservoir species. Synanthropic is a term that we use to name species that are associated with humans, but they are not domestic, like rats or feral cats or other raccoons, etc. Okay. Orthoptera is the other group with incomplete metamorphosis, also lots of species, not as many, but 13,000 species. 2 pairs of wings, the modification of the last pair of legs to produce sounds that they use for courtship behavior. They have a lot of modifications for chewing, and most of them are going to be, or no, all of them are herbivores. And one important feature about the orthopterans is that they compete for the food resources that we use. You have heard of how many crops that we grow are lost due to some species of orthopterans. So they produce, they have been responsible even for some famines in some parts of the world when there are these lobsters coming into huge swarms and eating all the crops. All right. Last thing that is important is that all the insecticides that we have used, most of the pest species in the orthopterans have evolved resistance to those chemicals. So those are bad news for us as well. All right. Questions? No? Ten more seconds. Okay. All right, next question. Ten more seconds. Okay. All right, questions? No? All right. The last large group of the animals, the Deuterostomia. And the Deuterostomia is going to include both invertebrates and all the vertebrates that we know of, including us. Okay? Now, the invertebrates are mostly the echinoderms, and there are a few chordates that are invertebrates that we will mention. I don't know if in this, no, not this chapter, the next one. Again, for you to remember, the Deuterostomia are going to have radial cleavage, the formation of the anus from the blastopore, and a lot of molecular evidence is the one that we use in order to determine that they are deuterostomes and the relationships between them, because you are going to see that some of them look something else other than chordates. We will see some pictures. But let's move into the first group of the Deuterostomia, the echinoderms. You know them well. They are the starfishes, and one of the main features of starfishes is that they have this water vascular system. So basically they use water to move things around their body, and also they use it to pump the water that they need in order to get the little legs that they have in every one of the extensions in the lower portion of the body, and that's the one that they use in order to move around. Now, they are considered to have radial symmetry, but they are not really radial, just as we are not bilateral, because if you cut yourself in half, not every half is going to look exactly as the other, because some of the organs are more on one side or the other. The same happens with echinoderms. Different organs are located in different portions of this radial body. They have separate sexes, and they are also going to be mostly predatory species. They are going to consume other species, and they are considered top predators and keystone species. We will talk about that in the last chunk of the course when we review ecology. And here a short video for you to see those feats that they use.
Animals in this phylum have a distinctive mechanism of locomotion. Within the sea star is an internal water vascular system that terminates in branches called tube feet. As the sea star moves, it uses chemicals first to attach each tube foot to the substrate and then to release each tube foot from the substrate. Water enters the water vascular system via the madrepore, which is a strainer-like structure on the dorsal side of the animal. This video shows what happens when a sea star is placed on its back in an aquarium. The tip of the sea star's arm can bend back on itself, and once the tube feet contact the substrate, the animal slowly pulls itself onto its correct side. Credit courtesy of Graham R. Kenton.
Okay. There are 5 main clades of these echinoderms. They are listed in here. And here is where we find the sea cucumbers. Sea cucumbers, even though they look more like worms, they belong to the echinoderms. And then the more classical stars are in the asteroidea, okay? And here we also have the sea orchins, which is the echinoidea. Questions? No? Okay, 10 more seconds. All right. All right, very quickly, a few things of these echinoderms. In the asteroidea is where you have the common sea stars, the ones that we observed the video from, and they are multi-predators, and they are going to use their very strong water vascular system and their strong arms and feet, tube feet, in order to open bivalves. So they are going to crack the shells by opening them, and then they will consume the animal inside those shells. And one interesting feature about them is that they can regrow portions of the body if they lose them to predators. Brittle stars, they are going to be different types of, depending on the species, they are going to be suspension feeders. They can also be detritivores. They can be predators or scavengers as well. This is how they look, the brittle stars. Then we move into the echinoidea, which is the sea orchins. And there are many species. They also have this feature of the water vascular system. They move, even though they look sessile. They use the tube feet in order to move around. This is a picture not of the most classical sea orchid. The classical sea orchids, they have these spikes. Probably you've seen them. Those are also mobile. They can move the spikes in order to move around their environment. Then we have the crinoidea, which are the sea lilies or feather stars. They have changed little since they appeared back in the Cambrian, so they have retained mostly that type of feature. Some of them can move using the extensions as legs, others are sessile, and they are going to be suspension feeders. Then we have the sea cucumbers. They are going to have these reduced endoskeletons, and they are not going to have these tube feet all over the body like the stars. They are going to have only 5 rows of tube feet in order to move around the environment. All right, we are moving into the chordates. Chordates means that there is this hollow tube that is going to develop during embryogenesis in the embryo, and is the tube where the nervous system of chordates is going to be located. In the case of vertebrates like us, that tube is the one that is going to become in our spinal cord, okay? Now, they have bilateral symmetry, and basically the chordates separated from the echinoderms about 500 million years ago. What period was that? About 500 million years ago. What was that period in the Paleozoic that is very important for life on the planet? A lot of species were produced.
Cambrian era?
Yeah, Cambrian period. Okay. Questions? Right, 10 more seconds. Okay. Right, a summary before we move into the next chapter. So we are dealing with animals. Remember the metazoa, all these listed here are metazoa. The ones at the base or the basal metazoan lineage are the sponges or porifera. Question for you, porifera sponges are considered to have true tissue or not? No, right? Okay. Now, after that, we arrive to the invertebrate animals that are considered with true tissue, the eumetazoa. But then here, the Cnidaria, which are the jellies, hydras, anemones, corals, etc., are they going to have the same number of tissues that we have as vertebrates? Or what is the difference in terms of tissue? Because they are considered to have true tissue now. But what is the difference between them and us in terms of tissues?
They have 2 joint layers.
Yeah, only two layers of tissue, okay? So they are known, the technical term is diploblastic, okay? And when we move away from the Cnidaria and then we arrive to the bilateria, which is all this group of animals that we have reviewed so far, we have 3 types of tissues, okay? And the technical term is tripoblastic. And then we have, within the bilateria, we have 3 main groups. Lophotrochozoans, so that includes the platyhelminths of flatworms, Syndermata, rotifers, and acanthocephalans. Within these 2 groups of Syndermata, which one only has parasitic species? Meaning the group contains only parasites. Within the Syndermata, what of these 2 groups are parasites? Yeah, acanthocephalans. Yeah, it's okay. Yeah, they are a mouthful. So, acanthocephalans, and they are also known as spiny-headed worms, because the head of those worms have a lot of different types of spines and things to attach to the host where they are going to be sucking food from it, blood, hemolymph, etc. Then we have the ectoprocta and brachiopoda, mollusks, which is a large group, and then the worms, the annelids. And then the dixozoans, we have 2 main groups, the nematodes and the arthropods, which is the largest group of animals. And now we start with the Deuterostomia, which we just reviewed the echinoderm. Now, next chapter, we are going to review the chordates, okay? Are there any questions? So these are beetles. Which of the insect groups is the most spacious group? That has a lot more species, the ones that contains more species. Coleoptera, which are the beetles, okay? And also they are very colorful, so they are very beautiful. But that is the most rich group of arthropods. Okay, let's take a short break, 3 minutes, so you can stretch before moving into the next chapter.
I only went because my dad was express. Okay. Okay. No point of me freaking going in my place. And I have so much work. Like, that person tried to properly move. Well, now my brother's getting tired. So my brother so I'm like, bro, I'm so ready for August. I'm so ready to get a bump because they okay. My
Alright guys, let's resume the lecture. Alright, last chapter of organisms that we are going to review before we move into ecology, the last chunk of the course. Alright, so what we have here is a phylogenetic tree of the chordates, and one general question about phylogenies. A phylogenetic tree is the truth about the relationships of organisms, and there is nothing to change it. So meaning it's grave on stone. No, right? So a phylogeny is always going to be a working hypothesis, because as we accumulate more evidence, some changes might occur in the phylogenetic tree that we currently have. Okay? So here we are going to start at the bottom with these chordates that actually don't have what we consider bones. They have some cartilaginous tissue and some calcium in some of the structures, but they are mostly soft tissue. Okay? Then we are going to have the development of jaws, and then mineralized skeleton, starting with the fishes, especially the sharks or chondroites. Then we are going to have low fins within the fishes, the coelacanths, which is actually the group where all the limbs that we possess started to appear, started to evolve in this group of low fin fishes. Then we are going to arrive to the groups of the tetrapods, where we have the limbs, okay, that includes the amphibians, and then we are going to arrive to most of the vertebrates that have an amniotic egg. Okay, so that's the tour that we are going to do about the chordates. About 60,000 species of vertebrates, so when you compare that number, it's tiny versus coleoptera, versus bacteria, versus many other groups in general like arthropods. So not that many vertebrates, but they include some of the largest and most dominant organisms of the planet. So they are all bilaterians, and they are going to comprise all the vertebrates, but there are two groups of invertebrates. The lancelets, that include the urochordates, and we are going to mention those two cases. So the urochordata and the cephalochordata are the two groups that are invertebrates within the chordates, okay? They have these two where nerves are going to develop, just like vertebrates, but they just don't have hard tissue, like bones, okay? And here is this phylogeny of the chordates. The sister group is the echinodermata that we reviewed earlier. Then we have the development of the notochord, which is a feature that we will see in a second, and then we have the cephalochordata over here, which is the lancelets as they are known. Then we have the urochordata, and then is when we jump into the vertebrates, where they have some kind of hard tissue. We will see all the details, and here on the right side you see the classification of the different groups depending on the traits that they have. Alright, so chordates. The main or the four key features for you to know that an animal is a chordate is that they have a notochord, they have the dorsal hollow nerve cord, okay, which is the chordates. That's where the name comes. They have pharyngeal slits or clefts, and then they start to have this radiated muscular tissue, especially in the rear portion that they use for movement. This is a diagram of a lancelet that has the 4 features even as they reach adulthood. Not every chordate is going to retain the 4 main traits when they are in an adult phase. Everyone goes through these 4 features when we start to develop as embryos, but depending on the species, we are going to lose most of those traits, okay? So here we have the hollow tube or the cord where all the nerve tissue is going to develop into the chordates. Then below that we have the notochord, which is more or less a solid structure giving support to the hollow nerve cord. Then we have the pharyngeal slits that are retained in many groups, and the function usually is to filter water for feeding, to capture particles. And then the muscle tail for movement. Okay, this is what I just mentioned. Now, most vertebrates as adults are going to have the muscle, of course, we need the muscles to move around, and they are going to be attached to the skeleton. And there are a few remnants of the notochord. The notochord is mostly lost in all vertebrates, or we don't have any notochord. Humans don't have the notochord. We notice the notochord only during embryonic development. The dorsal hollow nerve cord, I already mentioned this. The pharyngeal clefts or slits that they are going to use as a part of the mouth system where they are going to filter the water, and is going to be mostly in aquatic, for aquatic organisms, in particular for sessile ones, the urocordella that we will see in a second. Then the tail in most vertebrates is going to be lost, is going to be highly reduced in many vertebrates. All right, let's move to the different groups. Again, here we have the phylogeny, Cephalochordata, which is the lancelets, because of the shape that they have. And they are tiny, tiny organisms living in the bottom of water bodies. They look like this as adults. They are a few centimeters in length as adults, 6 centimeters, and they have all the 4 features of the chordates, okay? So what are the main 4 features again? Who would like to help repeat them?
Notochord.
Notochord.
Dorsal.
Dorsal hollow tube. Pharyngeal slits. And?
Muscular pulse.
Yeah, a muscular tail. Okay? So those are the main features. If you observe those 4, you know that you are talking about a chordate. Okay? Are there any questions? No? More seconds. Okay. All right, next group is the Urochordata, also known as tunicates. And the tunicates, when they are adults, they are going to be sessile, and they are going to be filter feeders. The 4 characteristics of the chordates in tunicates is going to be present only during embryonic development. Larvae are free living. They are going to move as part of the plankton in the aquatic environment, and then they are going to find a place where to settle. Here is a tunicate as a larvae. You can see that it resembles the lancelets. It has all the features of the chordates, and once they settle, they are going to become into something like this. So this thing here is more closely related to us than other animals. So if you look at an adult tunicate, you wouldn't think they are more closely related to us than other animals, like even the lancelet or the echinoderms. And they have all the features, so filter feeders. The pharyngeal slits are retained as part of the main digestive system, where they are going to filter the food that goes into the stomach, into the digestive system. And then they have the scoring siphon, and the anus portion drops all the waste material into that scoring siphon that is shown over here. Now, in terms of development, one interesting feature about the tunicates with all the other chordates is that they are going to have less number of Hox genes. And that might be related to the complexity of the organisms that we are going to review in this chapter. So when we are talking about chordates evolution, what we can see is that many of the same Hox genes are activated in the same portions of the embryo as the embryo starts to develop, even though they might end up looking very different. So for example, here, the brain of the lancelet is just going to be one bulge in the anterior end. It doesn't look like an actual head, yet the same type of Hox genes are going to be activated when you compare it with the brain of a vertebrate, okay? So many of the features that belong to the chordates are going to be apparent during embryonic development, okay? Questions? No? Well, talking about some important things of vertebrates, we have the Hox genes. Again, they are master genes responsible for the functioning of the well development of the embryo. So they regulate the processing of how the embryo is going to develop. And depending on the group of vertebrates, you are going to find just a gene or a few genes or a large family of genes that have been duplicated throughout the history of the chordates. And that enlargement of this type of Hox genes throughout the history of chordates is what it has allowed the development of more complexity in the body plans of the organisms, or the adaptations that we recognize in many of these organisms. Now, the neural crest is some specialized type of cells that is unique to vertebrates, and it shows up during embryonic development lining the edges of the closing neural tube. We are going to see some features, some diagrams in a second for this to be clear. Now, the neural crest cells are like totipotent cells. They are going to move to different portions of the developing embryo, and they are going to develop into different types of structures. Those structures can be even bones, some bones of the head of the vertebrate, some others are going to form teeth, some others are going to become nervous tissue, okay? Here is a diagram that is also in your textbook. So this is the closing neural tube during embryonic development. This is going to be the tube that eventually vertebrates is going to become the spinal cord that we all have. And the neural crest cells are the ones in green here lining this tube, okay? The notochord is shown in here because everyone has a notochord during embryonic development that gives support to the developing neural tube. And then as development continues, you have these neural crests that are going to migrate, and they are going to become part of the bones of the head or nervous tissue, or even internal tiny bones like the bones that we have in our ears, okay? That is a unique, neural crest cells are a unique feature of vertebrates. Alright, let's move into the next group that is known as the cyclostomes. Cyclostomes include these two groups, the Myxini and the Petromyxontia, which are basically, they already have some vertebrae, but they don't have bones or vertebrae as we know. So we have the hagfishes and the lampreys. So, and what they have is rudiments of these calcium hardened structures that we will see in a second. Now, one important feature of the cyclostomes is that they don't have jaws. Okay? In the evolution of chordates, all the way to this point, we still don't have jaws. Okay? They have a mouth, but they don't have jaws. Catfishes are all these aquatic. They have a little bit of cartilaginous tissue or hardened tissue in the skull. The adults are going to retain the notochord that is flexible and is made of cartilage. They have all the sensory organs in the head, but they don't have well-developed eye sensors. They use other sensors, and the mouth is going to contain these keratin structures that are like the teeth, but they are not actual teeth because they are made only of keratin. And they also have this line of glands across the body that are known as slime glands. And they are used to produce this slime that is used either for protection for the skin, and also when they are attacked by predators, they release these large amounts of slimes that are not highly toxic, but is going to give a bad time to the predator trying to eat them, because they are going to have a hard time removing the slime from the mouth. 30 species, well, at least we know 30 species, and all of them are marine. Now, the other group of the cyclostomes is the lampreys. They also don't have jaws. Remember, that's an important feature of cyclostomes. Lampreys and hagfishes don't have jaws. Okay? About 38 species, all of them are going to be aquatic, either marine or freshwater. And here we have a picture. Some of them are going to be parasitic, so they are going to attach to the skin of other fishes and basically suck blood. And this is one example of a mouth modified for sucking blood. So this example here is going to attach to the skin, and they develop all these type of teeth structures in order for attachment. Yeah, this is what I just mentioned. Now, when we are moving into early vertebrate evolution, the earliest fossil of a chordate is about 530 million years old. That belongs to the Cambrian period, the Cambrian explosion. And here are the imprints of those fossils. And the name, the genus that was given to that species was this one, and you can see that it is similar in body to the lancelets, okay? So it is a segmented body, and it seems to have a well-developed anterior portion, and it has the pharyngeal slits. So this is the earliest evidence that we have for the evolution of chordates, about 530 million years ago. Other extinct chordate are the conodonts, and they appear about 500 million years ago. They are known predators. They don't have jaws. They have many modifications of the mouth with this mineralized type of teeth and different types of teeth in order for capturing and processing the prey. They have large eyes, which suggests that they were in environments where there was not much light. Are there any questions? No? more seconds. Okay. All right, we continue with jawless vertebrates, and these are other examples of extinct animals, and they larger and better protected jawless chordates, and they are considered vertebrates, appear later on in about 485 359 million years ago. That is between the Endordovician, Silurian, and Devonian periods, and these ones are believed to have gone extinct during the Devonian. The Devonian is about 410 million years ago, all the way to 350 million years ago before the Carboniferous period. So one thing that they have is a very well developed pharynx, very muscular pharynx that they use to process the prey that they capture. They have some portions of the body that were mineralized, so they had some kind of bone structure already in their bodies. Now, early vertebrate skeletons, or what we consider vertebrates, contain also portions of cartilage, which is it's a portion of hard tissue that doesn't have minerals like the bones. Now, mineralized bones, like the ones that we have, appeared about 470 million years ago. And the actual type of skeletons, what we know as a skeleton, like the ones that we have, appeared 430 million years ago. That is by the end of the Silurian period in the Paleozoic era. Okay, so those are a few features that we are more or less confident of when cartilage and bones, mineralized cartilage and bones started to appear. Okay, natostomes. We were talking about jawless vertebrates. The living two groups are the cyclostomes, which are sister species, which are the hagfishes and the lampreys. Now we are moving into the natostomes, and natostomes is the technical term for all the vertebrate organisms that have jaws. Here now we are jumping into fishes, and the oldest fishes or the basal lineages of natostomes are going to be the sharks and their relatives, okay? Derived characters of the natostomes is the jaws, and they are hinged jaws, and there are many modifications of the gill slits. Those gill slits that the lanceolets use for filter feeding, many of those slits, the support structures for those slits were modified into bones of the jaws. Here is a little picture diagram that I think is also in your book, where you have the gill slits and then the skeletal rods that give support to this in more recent vertebrates, and some of the more anterior skeletal rods are the ones that were modified to give rise to the bone of the jaws. Okay. Other important characters of natostomes or jaw vertebrates is a lot of genome duplication of what? The Hox genes. Hox genes, we said again, they are very important for what reason?
Diversification.
Say it again?
Diversification.
Yes, of course, they are correlated with diversification, but what type of genes are they and why they are important? During development, yes. What they do during development? Yes. So they are the master genes that coordinate the functioning of the genome during the development of the embryo. Basically, they coordinate what other genes are activated or suppressed depending on the stage of development of the embryo. Okay, that's why they are very important. And again, in jaw vertebrates, they got duplicated more than once. So we have, as humans, as primates, we have a lot of Hox genes. And other important feature is the enlargement of the forebrain. So it's when we start to see in earnest the central command of a nervous system in the anterior portion of the body. We have, depending on the species and the group, enhanced senses for different purposes. It can be olfaction, it can be vision. In our case, it's very important the vision, but if you look at the brain, for example, of a dolphin or even of a bat, the more enlarged portions of the brain are not going to be the portions of our brain, which are the frontal lobes. Those are going to be different portions of the brain. In the case of bats and dolphins, it's going to be the hearing portions of the brain that are more acute, okay? Lateral line system is basically of aquatic species, is a sense organ that goes along the body of the aquatic animals, like in fishes. Whenever you have a fish or you buy fresh fishes that you cook, look at the skin, you are going to see that lateral bands that are openings. There are little holes across along the body, and they are a sensory organ. They perceive movements and other electrical fields of their aquatic environment, and then they send the signals to the brain to process the information. That's the reason why many, for example, sharks, that's how they detect many prey species. They detect the electrical movements of the fish moving around them, and they use a lot the lateral line system. In terms of the fossils of jaw vertebrates, they started to appear about 440 million years ago. That's the end of the Silurian and the beginning, end of the Silurian, because the Devonian starts in 440 million years ago, more or less. And some other adaptations for the aquatic species that are the first ones to evolve in terms of vertebrates is all the fins that you know from the fishes, the paired fins, the tails, the dorsal and ventral fins, that eventually they are going to be co-opted into structures that are going to start getting calcified and develop like into walking appendages. Okay, we will reach that part. Now, there are modifications in the bladder. The bladder is used in many fishes as a buoyancy apparatus to maintain buoyancy when they are swimming or when they are resting. Depending on the species, there is going to be the modification, and some modifications of the bladder is going to be used also as an extra lung. So they use it to be able to breathe air. Like for example, here in the Gulf of Mexico, we have the garfishes. Anybody knows garfishes? Yeah, garfishes is a fish. It's a very ancient lineage of fishes. They have the head of a crocodile or an alligator, and then the body of a fish, but they are covered with these calcium scales. So it's like an armor. The body is like an armor all across the body. But they can breathe air. Okay, so they can of course breathe below water, but when there is very low oxygen in the water, they can come out and breathe air. Questions? Okay? All right, talking about fossil natosome or jaw vertebrates, one of the earlier groups in terms of fishes was the placoderms. And they basically were like armored fishes that range in size between 1 meter all the way to 10 meters, and they looked something like this. So they were, of course, predators in water systems. There is another group of these early fishes that is known as the acanthodians, and they lived between 444 million years ago into the Devonian. 444 is the Silurian, and then here they went extinct during the Devonian period. By the end of the Silurian, early Devonian, 420 million years ago, is when we start to see the appearance or the divergence in the aquatic environment of the fishes that were the ancestors of current day fishes that we have in the oceans. We have those three groups listed in there. The chondroichthyans, which is basically sharks, rays, ray fin fishes, which are the ones that you have for dinner most of the time, and then low fins, which are the coelacanths, which are the ancestors of tetrapods. We will see those groups. Are there any questions? No? Okay. All right. Let's move then to the next group, the chondroichthyans. Now we said here, what if we put a trait that is shared by chondroichthyans and the rest of the vertebrates here in the common ancestor, what would that be? If we compare them to the cyclostome group? Jaws, okay? Right here is the evolution of jaws. Now we're moving into the most basal group of jawed vertebrates or natostomes, which are the chondroichthyans, sharks, rays, and basically here, it's mostly cartilage, the skeleton of these animals, but we start to see some calcium impregnations into some portions of the cartilaginous skeleton of these organisms. We see some impregnation of calcium in the scales, also in some of the vertebrae, but nothing like our bones just yet, okay? In terms of chondroichthyans, we have about 1,000 species, and some of those species are known to the less known group known as the chimeras. So these are also like related to sharks, but they are mostly benthic organisms that are not as well known as rays or sharks. Okay. One important feature of sharks is that in the bladder they have oil, and also in the liver they store a lot of oil, and that allows them to give some buoyancy even when they are resting. When they are resting, they need to keep some buoyancy, and they also need constant movement, okay, because they need to be moving or creating a current into the gills to keep breathing air. So they are never completely at full rest. This is what I just mentioned. The largest sharks are actually not predators. The whale shark actually eats tiny arthropods. Anybody remembers what is the arthropod group? I just mentioned a few minutes ago. It belongs to the plankton, and the whale shark eats that shrimp-like crustacean. Krill. Okay, krill again is very important for the food chains in the aquatic environment, and the largest shark depends on it. But then most sharks are going to be top predators, carnivores. One feature is that as carnivores they have a short digestive system, yet at the end of the digestive system in the stomach they have this coil area that is going to be helpful to slow down the passage of food so they can take advantage of the food that they are consuming. And of course they have very acute senses of smell and sight. They have very good eyes. Okay, I'm going to finish just with this. In sharks we can find 3 types of reproduction in how they produce the babies. They can be oviparous, ovoviviparous, and viviparous. Oviparous, they lay eggs. Ovoviviparous, they produce eggs, but the eggs hatch inside the body of the female, and then the babies are released after that. Viviparous, similar to us, the babies are developed in the uterus. They are nourished through something like the yolk that you will find in eggs, and then they can also feed on other eggs that are produced in the uterus of the female, but they are born alive. Okay. All right. See you Thursday, guys.